Dear students, welcome to PUC second year biology class. We are studying the chapter Principles of Inheritance and Variation. In that, some of the Mendelian disorders we are studying. In that, the first disorder called the hemophilia, we have studied about that. And the second type of disorder is color blindness. It is a sex-linked or the X-linked recessive disorder due to defect in either red or green cone of eye. So it is default of that gene. It results in the failure to discriminate between the red and green color. The person will not be able to differentiate the color red and green. So this is what the color blindness. It is one of the sex-linked recessive disorder due to defect in either red or green cone of the eye, which results the person will not be able to differentiate or distinguish red and green color. It is due to mutation in some genes in the X chromosome. So the chromosome which is responsible for this will be missing. Therefore, the person will not be able to differentiate, distinguish, or identify exactly the red and green color. It occurs in 8% of males and only 0.4% of the females. It is because the genes are on the X chromosomes. Genes are present on the X chromosomes. So it can occur more in the males compared to the females. Normal allele is dominant and the recessive allele causes the color blindness. The recessive allele is causing the color blindness. The son of the heterozygous woman carrier has 50% chance of being blind. A daughter will not be colorblind unless her mother is at least a carrier and her father is colorblind. So it very rarely occurs to the daughter, you can say. This is one of the disorder called the color blindness. And another one is sickle cell anemia. This is an autosome linked recessive trait. It can be transmitted from parents to the offspring when both the parents are carrier for the gene or heterozygous. If the parents are carrier, and this disorder can be passed on to the offsprings. The disease is controlled by a pair of allele called HBA and HBS. Homozygous dominant, if it is there, then they will be normal. Heterozygous carrier are there, then that leads to sickle cell trait. And homozygous recessive, if it is present, then obviously the person is getting affected with this type of disorder called the sickle cell anemia. The disease is caused by substitution of glutamic acid by valine at the 6th position of the beta globin chain of hemoglobin. On the hemoglobin chain, it is substituted with glutamic acid by valine. This is due to single base substitution at the 6th codon of the beta globin gene from the glutamic acid to valine, which is happening here. So the mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC. The RBC shape will change from biconcave disc to the elongated sickle cell structure. You can see here. This will be the normal hemoglobin, which is biconcave. But if it is getting affected, then it will become elongated, which we call it as sickle cell. Sickle-like structure is being appeared. It is because here, if it is normal, glutamic acid is there, but this glutamic acid is replaced by the valine. At the hemoglobin, if glutamic acid, this is being glutamic acid is replaced with the valine at the sixth position. You can see sixth position. 1, 2, 3, 4, 5, sixth position. At the sixth position, if glutamic acid is replaced with the valine, then that leads to the sickle cell anemia. This is one of the Mendelian disorder called the sickle cell anemia. Again, it is shown here. You can see 1, 2, 3, 4, 5, 6. At the sixth position, glutamic acid is there. That means it is a normal. If it is affected with the sickle cell anemia, that means glutamic acid is being replaced with the valine. If it is replaced with the valine, then person, you can say, is having the sickle cell anemia. Another type of disorder is called phenylketonuria. Mendelian disorder is phenylketonuria. An inborn error of metabolism. It is an inborn error of metabolism, autosomal recessive trait. Because of the autosomal recessive trait, the phenylketonuria occurs. It is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. It is caused by the mutation of the gene that codes the enzyme called phenylalanine hydroxylase. Single gene mutation is undergoing the changes. The affected individual lacks this type of enzyme that converts the amino acid phenylalanine into tyrosine. As a result, phenylalanine accumulates and converts into phenylpyruvic acid and other derivatives. So in our body, that was the mechanism, metabolic activity which was carried out. But because of the absence of the enzyme, they accumulate in the brain, resulting this phenylpyruvic acid starts accumulating in the brain. That results the mental retardation of the person. Because the enzyme is there, phenylalanine hydroxylase enzyme is there, therefore that will convert the amino acid phenylalanine into tyrosine. But as a result, phenylalanine accumulates. Tyrosine, when it is converted, the phenylalanine accumulates and converts into phenylpyruvic acid. Because this phenylpyruvic acid starts accumulating in the brain, that affects the brain, the person will become mentally retarded. These are also excreted through the urine because of poor absorption by kidneys. There is possibility of that also. So this is one of the disorder called phenylketonuria. Thalassemia is another type of disorder. It is an autosome linked recessive blood disease. It is an autosome linked recessive blood disease. It is transmitted from parents to the offspring where both the parents are unaffected carrier for the gene. If it is heterozygous gene is present, then this type of disorder is occurring in the human being. It is due to mutation or deletion. Mutation, if it is undergoing the changes or if it is deleting from that position, it results the thalassemia. It results in the reduced synthesis of one of the globin chain, alpha and beta chain that make up hemoglobin, which is responsible for making the hemoglobin is affected. This forms abnormal hemoglobin molecules resulting into anemia. So abnormal hemoglobin molecules will result in the anemia. Thalassemia differs from sickle cell anemia. Thalassemia is a quantitative problem, synthesis of few globin molecules. Sickle cell anemia is a qualitative. It is a qualitative problem, synthesis of an incorrectly functioning globin occurs. Synthesis of incorrectly functioning globin is taking place in the sickle cell anemia, but in thalassemia it is a quantitative problem, synthesis of few globin molecules. This is another type of disorder called thalassemia. Another type of Mendelian disorder in this thalassemia, there are two types, alpha thalassemia, beta thalassemia. Here, production of alpha globin chain is affected. It is controlled by two closely linked genes called HBA1, HBA2 on chromosome 16th of each parent. On the chromosome 16th, these genes are present. It is due to mutation or deletion. If it is undergone the changes or if it is deleted of one or more of the 4 genes, the more genes affected, the less alpha globin molecules are produced. If it is affected, alpha molecules are being produced very less. Same with the beta thalassemia. Here production of beta globin chain is affected. It is controlled by a single gene HBB on chromosomes 11th, which was present on each of the parents. It occurs due to mutation of the gene. Both the genes are there. Now, chromosomal disorders. Some of the chromosomal disorders you will be studying, and those chromosomal disorders you'll see in the next classes.